All right, so in the last PowerPoint slide, we saw that in the Miller-Urey experiment, they were able to abiotically synthesize some of the monomers that would be necessary for life to get started. And they did so using, some of the things that they thought would have been in the early Earth's atmosphere and an energy source that, as we said, could have been, volcanic in nature, could have been lightning, could have been UV radiation. But out of that, we got all these monomers. Now, just forming things like nucleotides or ATP or amino acids or glucose, none of those things by themselves can really get us to the point where we have something living. We have to take those monomers and turn them into polymers. And that's what the Fox experiment showed us that was possible. Basically, what they did is they put all these monomers that would have come out of the Miller-Urey experiment, and they would have put them into clay and hot sand. And with the pressure being exerted on those uh, molecules, in addition to the heat, then you could have the formation of the polymers that would be necessary for life. So we're talking about forming proteins, um, nucleotides coming together and forming nucleic acids that are long, like DNA or RNA. And so this is the sort of next step in the formation of something that's living, is that we've got to have these polymers. Once you get the polymers, now you need to be able to have an environment that's enclosed and is different from what's going on in the outside world. And that's where the Oparin experiment, where basically what they did was they put phospholipids and some of these other polymers into a mixture in a container. And basically what happened was, in the presence of water, these molecules formed membranes, just like we would see in a regular cell. So once you have all of these polymers, what it's saying is that you could have randomly, without any sort of external influence, the formation of things that would have looked similar to cells but wouldn't have had the replication machinery in place. So they would have been able to control their internal environment um, compared to what was going on the outside. And so that was another important experiment, just showing us how that process could have happened. How do you get something that looks like a cell even though it might not be able to replicate? So what are we missing? Well, in our definition of life, you need to be able to replicate your genetic material and pass that down onto your offspring. And that's where the next slide comes in terms of its importance. We now think that RNA was probably the first genetic material, not DNA. And the reason for this is that scientists have found that in a laboratory, RNA can carry out enzymatic functions. And so we call these RNA sequences that can do certain enzymatic functions, we call them ribozymes. And the idea is that some of these RNA molecules would have had enzymes that would have allowed them to replicate themselves. And once you have a sequence of RNA that can replicate itself, now you could have it inside of one of these protobionts, one of these early cells, and potentially, you know, have the RNA that's inside of that protobiont. If the RNA replicates itself and then the protobiont were to split, then you would have life being created, where you'd have some sort of genetic material that could be passed on to subsequent offsprings. And the other reason why we think that RNA was a good model for the very first genetic material was that in the laboratory we've seen that these RNA sequences that can replicate themselves have almost sort of like natural selection on a molecular level, where those sequences that are best able to copy each other are the ones that are going to produce more copies, and sort of the whole idea with evolution where the greater your chance of survival, the more opportunities you have to mate, the more offspring you can produce, and the more of your genes you can pass on. And so it's the same thing here that, the better they are at copying themselves, the more of their genes you'll find in the subsequent generations. Um, so I'm just trying to think of if there's anything else I want to add. Oh, at some point, we know that RNA as our original genetic material had to be transferred into a DNA form. And if you go back and remember what we had talked about with viruses, that could be the origins of an enzyme like RNA transcriptase. And in fact, we do think that viruses probably played an important role in um, sort of how do these RNA sequences get into protobionts. Um, we know that viruses can inject their genetic material into host cells. Were viruses, going around in the early Earth's history and injecting their what do you call it, their genetic material into these protobionts? Is that how life got started? Only because of what viruses were doing? And so that's sort of an interesting question. The key here at this point is that we don't really know how all of these parts of the process happened. But from what we've seen in the laboratory, what we can say is that there was a way. There was a way that without intervention of any sort of supreme or divine being, we were able to get the very first life forms. It was all without intervention of any other living thing. And that in many of these cases, what we're finding is that, different labs are working on different pieces of this puzzle. And we're finding that, the original formation of life probably could have happened in many different ways. And so that's sort of the take-home point out of this, is that we may not be entirely sure, but we can be sure that it was able to occur with very little intervention uh, from some other person or some other being. So there's a bunch of different theories on where did life first get started, and one of the theories is that it got started on the surface of the Earth. Some of the evidence in favor of this was that UV light or lightning could have provided the energy for some of those chemical reactions that we were just talking about. That there was a no oxygen atmosphere, it was a reducing atmosphere, and that favors certain chemical reactions. The Miller-Urey and Fox experiments were both dealing with what could have happened on the surface of the Earth. And remember that our very first fossils are stromatolites, which were in those intertidal pools, and that's on the surface of the Earth. The problem is that we think that at the beginning of Earth's history, the sun was much weaker than it is now, and that means that the surface of the Earth probably would have been covered in ice. And obviously, if it was covered in ice, that's not really a great place for life to get started. But then there's another problem as well, which is that nobody really knows what the composition of the early Earth's atmosphere was. We can guess that there was methane and that there was ammonia, but nobody's really certain. Deep sea vents are a pretty good idea, where we think that, I think that probably the consensus is that this is where life got its start. The reason is that in these vents there's lots of important molecules that leave. So unlike being on the surface of the Earth, where there would have had to have been the right mixture of gases, different vents would have had different mixtures of gases, and they would have all been contributing something to the water in the area that would have supported the formation of these molecules. And we can be certain of that because we know what's coming out of these vents today, and that provides a little bit more certainty. Also, you have the energy for chemical reactions coming from the vents. Some of these vents even release light. So there's heat energy, there's light energy, and that could have supported life or the synthesis of these molecules in these areas, and therefore the formation of life. Also, some of the oldest fossils that we have are archaea, and archaea is this group of prokaryotes that um, currently live around these deep sea vents. And so we think, well, if some of our first fossils of our archaea, then it could have started there as well. So the problem is that it's really hard for us to imagine that life would have begun in some of the most extreme environments on Earth, but it's actually this idea that has led us to believe that there might be life on some of our moons around Jupiter or Saturn, where there's ice covering a liquid ocean underneath, and that sort of gives us hope that there might be other life forms in the solar system itself, let alone the entire universe. Outer space is another theory of where life's got to start. We do have an asteroid 4.5 billion years ago that has 80 different amino acids, and they are not contaminated, meaning that those amino acids are not naturally found here on Earth. So we know that they came from the asteroid itself. And we also know that in early Earth's history there was many asteroids and comets hitting the Earth's surface, which is why we have oceans. Remember that the oceans were formed by ice comets. The problem with this is that you know that when things come through our atmosphere, they heat up, and most of the objects that enter our Earth's atmosphere are vaporized, and nothing hits the Earth's surface. There's kind of a funny Simpsons where there's a gigantic asteroid heading for the town of Springfield, and then by the time it gets there, it turns into a little rock and hits Hans Moleman on the head. So kind of gives you an idea. The likelihood that something would have survived impact, very, very small. And we've also never found life on another planet, so that makes it less likely that life would have been started from outer space. So we have all the conditions here on Earth, and we think that it probably was at the very least here on Earth and most likely inside of the deep sea vents.